Good morning, everybody. Glad to see so many of you here for this graduate research showcase. My name is Daniel Kirsch. I'm the Associate Chair for Education, filling in for Professor Mari Ostendorf, who is unfortunately not able to be here. My job this morning is very simple. I just need to introduce Sumit and Momona, who have put this showcase together and who will be taking care of hosting the ceremony. Thank you.
Thanks. Hi, everyone. Thank you for joining us for our second annual Graduate Research Showcase, where we highlight EE student, graduate student research and innovation. Today, we will have 5-minute talks by 8 speakers from very different areas. We will take one quick question at the end of each presentation. If you have more questions, please ask the presenters at the end of the colloquium so we can get through all the presentations. We will have voting at the end for the $1,000 travel award, so please think about who you want your vote to go to. Thanks.
Okay, so to begin off today's talk, we have Sandamali Devaditya from the lab of Dr. Matt Reynolds.
Hi, everyone. Okay. I'm Sandamali, and I work with Professor Matt Reynolds. And today my talk is on accelerated millimeter wave image reconstruction. So my research is broadly on applying computational imaging to improve millimeter wave synthetic aperture radar. And yeah, so how many of you like that long line at the airport security? Okay, you have to remove your shoes, take off your belt, and then go to the scanner, and then you have to stand like this. And this scanner uses millimeter waves to image your body. Okay, and then the security person says, okay, unless, of course, you are carrying a bomb, and we get the results in almost real time. Okay? So what's stopping us from getting results even faster? The problem is, if you want images really fast, we have to wait, you're not going to get good quality images, because image reconstruction is a computationally complex process. If you want really good images, like, for example, in this movie, Total Recall, you have to wait some time to get those images. So can we obtain images faster and still have high resolution? So that's the question I'm asking. So let's move on to the details. So this is a system model of a synthetic aperture radar. So we have an antenna which is moving, and while moving, it scans the scene. So it transmits a pulse which hits an object, and the reflection is received back. So at the airport scanner, the thing that goes swish around you does the same thing. So at each position, we get the measurements, and we can concatenate them in a matrix like this. So here, Y is the received signals, X is the reflectivity of the scene, and H is the measurement matrix, which gives the relationship between X and Y. We need to estimate X, and for that, we have to find the inverse of H. But the problem is, you can't find the exact inverse. So a best solution would be a pseudo-inverse or a least square solution, but that's computationally complex, and it's mostly ill-posed. So in practice, we rely on approximation methods, like a match filter or the range Doppler algorithm. But the problem with this is that the resolution is limited. And also, if you are imaging a large 3D image with lots of, billions of voxels, it's going to take a lot of time. So this is my solution, a partition inverse algorithm. So we first partition the measurement vector Y into subvectors based on the position of the antenna. And then at each position, we only consider the part of the scene which is illuminated by the antenna. Okay? And so now we have the overlapping partition scenes, the X tildes. And our H is a block diagonal matrix of HS. So this HS is a submeasurement matrix. And because of the geometry, because the antenna looks at the same direction at all positions, this HS is a constant. Okay? So now we can find the inverse using a block diagonal matrix of the pseudo-inverse of HS. And we can easily implement this in parallel. So this is the block diagram of the implementation. And we can use a graphic processing card to speed up the process, and it's faster than even the match filter. Okay? And since we are using the pseudo-inverse and not the match filter, even the resolution can be improved. So this is an experiment we did in the lab using K-band horn antennas. So we are imaging this small balloon dog here. And with the mesh filter, it took us 3.5 hours. And with the partition inverse, we could do it in less than a minute. So we had a speed of 250 times for this. And also in our simulations, we found that the resolution is improved by 24%, even at 0 dB SNR. So now we have an algorithm which is robust to noise, which is fast, and which has better resolution. Okay? So with that, I'd like to conclude. And you can refer to these publications based on this work for more information. And this UWEE you have been seen throughout the slides. It's also a millimeter wave image we did in the lab using dynamic metamaterial antennas. So thank you, and I'd like to answer a question.
Questions? Does anyone have any questions? All right. So next we have Miles Gander from the Clavens lab.
Hi everyone, my name is Miles, and like Sumit said, I'm from the Clavens lab, and today I'm going to talk to you guys about some computational work that we do in the Clavens lab that's a little bit off the beaten path of what you normally see in an electrical engineering department. And that is namely making digital logic circuits inside of eukaryotic cells, in this case actually inside of yeast using DNA circuits. So as we all know, digital logic and electronics is extremely important, and it allows us to take simple logical gates, create more complex circuits using these gates. You can implement them in transistors and then make some pretty incredible computational devices that are really useful all across all of our lives every day. So if you kind of squint and turn your head and look at biology the right way, actually biology performs some digital logic as well. And my favorite example is cellular differentiation after fertilization of a human embryo. So based on this one program, this genetic program, which is the genome, based on environmental factors and circuit inducers, the cell makes decisions about what tissue type to differentiate into. And so from one set of DNA sequence, you end up getting all the sorts of different tissue types that make up you and me. And so this is arguably the most powerful process in biology because it creates us. So how could we recapitulate this kind of behavior where we're taking information in, making a decision, and actuating some biological output? So what we did in the Clavens lab to try and attempt to recapitulate this kind of behavior is using a gene wiring system with this dCas9 protein. So this dCas9 protein is essentially a protein that is guided by this small guide RNA down here. And this guide RNA has a specific identifier tag that can be located to a specific gene, and if it binds to that gene, can turn it off. So in a way, this is sort of an inverter whose input is the guide RNA and whose output stage is this gene, and by binding to it, can turn it off. So what you can do is actually come up with genes that have two binding sites for two different guide RNAs and whose output is the guide RNA. And in essence, you get a NOR gate. And this NOR gate is just a small piece of DNA, and you can integrate it into cells, and you can actually wire them up together. And this is what we've done. So I'm going to stress that this is actually really difficult to do in biology, and these are among some of the largest circuits that anyone has actually able to implement as of right now. And I'll explain these plots here. So basically, you have a fluorescence count of individual cells, and so these circuits are expressing a fluorescent marker that you can measure with flow cytometry. And you can see the two inputs are indicated by the plus and minus signs on the side, and by wiring up these NOR gates, we're actually able to create these different two-input logical systems. And so the goal of this is to try and build up these circuits so that we can get more complicated function. These are among some of the biggest, but we wanted to ask, okay, what's the simplest way that we could build up these circuits to see how large of this architecture we could make? And the most simple-minded way to do that is these repression cascades. So you start off basically with just one gene expressing the fluorescent marker. You add an inverter, the signal goes down. You add another one, it goes up. Another one goes down. And at this point, this was the largest repression cascade ever made. So we were really excited, and we wanted to see how many more can we go. So we got to 5, 6, and then 7, and then 8. And so this, or 7 total guide RNAs, but 8 different circuits. And so this was really exciting. This is the largest eukaryotic gene circuit that's ever been published. And hopefully, by demonstrating that we can build circuits of this size, we'll actually be able to move to more interesting computation in the future. So like we were saying, you can take these simple logical gates and combine them up to make computational devices. And in the same way, you can use genes, build them up into gene circuits, and then create tissues that are capable of useful things. We have a tree here as an example, but you can imagine using any other sort of agriculture or things like that. So summary, future work. I've showed you a NOR gate architecture based on DCAS9 protein. We created some large circuits with these and some repression cascades. And the remaining questions are, how can we increase the complexity of these devices? What are the immediate applications? So what can we actually control right away with these things? And if we add feedback, can we actually create some memory circuits that could have even more improved complex function? So with that, I'd like to thank my co-authors, Justin Vrana, Willie Voje, my advisor, Professor Clavens, and my collaborator and kind of co-advisor on this work, Professor James Carruthers from Chemical Engineering. So thanks for your attention, and be happy to answer a question.
So during normal computation, you use up voltage. In this type of computation, what resource is getting consumed, and how do you refresh it?
So the resource getting consumed is actually the guide RNAs being transcribed from these genes. And the genes are set up to actually constantly express, so it's sort of like a background voltage. So these guide RNAs are always existing, except when Cas9 is bound to the gene and has it turned off. So the voltage, in a sense, is the guide RNAs.
Okay, so thank you, Miles. Next, we have Apurva Sharma from Dr. Matt Reynolds' lab.
Hello everyone, I'm Apurva Sharma, and today I'm going to talk about implantable antennas in biomedical devices. Imagine moving a mechanical arm just by thinking, or playing video games with your mind. This may seem like science fiction, but engineers and scientists are trying to make it possible, and the technology is called as brain-computer interface. So in brain-computer interface, computer acquires a neural signal, analyzes the data, and translates the signal back to brain with specified signal. And the problem with current brain-computer interface is shown in the figure. So on the top of head, it requires big amplifier, and those amplifiers are connected to cables. And such kind of wired technology is not ideal. So we are trying to design implantable wireless devices for neural recording and stimulation. And to make a wired system, antennas play an important role. And it is a challenging task to design implantable antennas because of limited volume, limited power budget, and need for high bandwidth. So here I am presenting dual band antenna system, which has 2 antennas in the implant board, and we have recently filed a patent on this architecture. As shown in the block diagram, I have 2 devices, external device, which is going to be in close proximity to human tissue, and the implant device approximately 1 centimeters below from skin surface. And both devices have 2 co-located antennas, one designed at HF band and another designed at UHF band. So as shown in the diagram, external antenna, HF transmitted antenna is used to supply power to implant via inductive coupling, and UHF antennas are designed for data communication. Now the question is, why we are using 2 frequency bands? Why not single frequency band and make implant to be simpler? It's because HF band has a limited bandwidth, which is 14 kilohertz, and it cannot support high data rate. While UHF band has a wide bandwidth, and for recording and stimulation, we need more number of channels and high data rate. But the trade-off is higher RF absorption in the tissue as compared to HF band. So by designing dual band antenna system, we can efficiently transmit power as well as data communication at high data rate. Let's talk about specific absorption rate. It is the rate at which energy is absorbed by human body from RF exposure. And it is a very critical parameter if you are designing antennas because it is not allowed to increase tissue temperature by more than 1 or 2 degrees Celsius, which can cause heat exhaustion, heat stroke, and ultimately it can damage human tissue. So our dual band antenna system has a low specific absorption rate as compared to regulatory limit in USA. So to check the performance of antennas, we did various testing using different environment. For example, one using saline solution, which is salt and water solution, and it serves as a tissue proxy, and another using chicken tissue experiment. So in the second figure, there is a chicken, and the implant is inside the chicken tissue. And we found HF power efficiency to be 17%, which is sufficient to power up our UHF board. And UHF communication link loss from implant to external side, we found 7 dB higher in the case of saline as compared to chicken tissue. This is because saline has high loss tangent as compared to chicken, which means most of the field is going to be absorbed in the saline as compared to chicken or any other animal tissue. So if a system is working in saline, means your system should work in animal tissue. And at the end, I would like to say thanks to CSNE for giving such a nice project, and Professor Matt Reynolds. And if you are interested, you can look at our journal paper, and recently our conference paper got nominated for Best Paper Award, and the results are going to come tomorrow. So that's all. Thank you. Questions?
Questions? Doesn't seem like we have any questions.
Thank
Okay.
you.
So the next speaker is Ethan Keeler from the Photonics Lab.
Thank you. So today I'm going to talk about my research group's work on a MEMS resonant mass sensing platform for the integration of optical trapping, ultimately to do biological sensing and long-term monitoring of single cells. So in biology and medicine, for a very long time, we've known how to fully characterize tissues and fluids and try to understand what happens in living organisms. What has been highly challenging and we haven't fully characterized is what's happening on a single cellular level and looking at properties like mass, density, and stiffness, and trying to achieve resolution on such a small size scale, which can be 10 to the minus 12 grams. And by doing that, we can answer some very fundamental questions, such as whether the size of cells affects their growth rate, how those cells react to various stimuli, so therapeutics and doing drug discovery. You can look at how those cells interact with each other, or whether the cells are internally regulated. And then we can look at things like what happens in cancer, where cells become deregulated and have these kind of uncontrolled growth modalities, and how we can intervene in these situations. And so you might wonder how we even measure such small masses of single cells. And it turns out that you can use MEMS resonators. So here is pictured a cantilever structure. So it's a beam structure that's fixed on one end, and it has a fluidic structure, a fluidic channel integrated. And the structure will vibrate at some natural resonant frequency, which is a function of its mass and its dynamics. And if we flow cells or particles into the structure, we perturb its mass, and we can change that natural frequency at which it vibrates. And we can monitor a shift in this resonant frequency. And by observing this shift, we can determine properties about the cells, these very, very small, precise properties. And by flowing cells in and out of these structures, we can monitor them over time and determine some of the dynamics of what's happening in the cell to various stimuli or various drug flows. But it turns out with these devices, there's one fundamental problem, and that is that in MEMS resonators, they're not only highly sensitive to very small masses, they're also highly sensitive to where those masses are placed on the structure. And so in these kind of fluidic environments where there's quite a bit of variability in the trajectory of these particles isn't always well known, you can get a lot of error accumulation in the output measurement of your device, which is quite challenging when you're looking at such small properties on a single cell scale. And so in our research group, what we're working on is integrating optical trapping with these MEMS resonators. So in optical trapping, we can very precisely position and fix particles on these structures. So this is an illustration of our devices showing a bridge-like resonator, and we're using a laser beam to fix and position cells on top of the structure to deal with some of this variability. And underneath the cell, you'll see this kind of patterning, and that's very important because in optical trapping, what you're using is a very highly focused laser beam, and you're focusing it on the cell. What ends up happening, and quite rapidly, you can heat the cell up and kill the cell. And so it's membranal burst, and you perform opticution in a sense. And so in our lab, what we've shown is that by structuring the surface, creating these kind of grooves, we can enhance the trapping pattern and decrease the required intensity and increase the force on these cells. And so we can trap cells for long periods of time in these devices by using this technique and ensure good coupling between the substrate and the cell itself. And so we've been able to simulate these devices to validate the need for this positional accuracy in these MEMS resonator devices and validate the need for optical trapping. We've looked at the possible resolution of these devices, which is on the 10 to the minus 15 gram range, which is quite adequate for looking at how cells would change and vary with drug discovery or various stimuli. We have fabricated these devices here on campus in the Washington Nano Fabrication Facility. So this is kind of the overview of our fabrication process, but I'll spare you the details. And these are some images of devices that we fabricated. So for clarity, on top is the bridge-like resonating structure with these grooves that I've been mentioning. And on the bottom is the fully integrated device with a fluidic channel. And on an end note, I just wanted to show a video of optical trapping in these structures because it's kind of cool and exciting. So here what we have is a particle that's trapped, and we're manipulating it along the channel, and we're going around various obstacles with and against the flow of fluid, picking up various particles. And so that's using a laser beam to trap those particles in place of the channel on these structures. And of course, none of this would be possible without a great research lab. So here is pictured the Professor Lin's Photonics Research Lab, and my contact information in case you have any questions, and I'd like to thank you for your time.
Questions? No. All right. Thanks. So up next, we have Farah Nadeem from Dr. Mari Ostendorf's lab.
Hello, everyone. My name is Farah, and I'm with the Tile Lab. And today I'm going to talk a little bit about my work on building a text classifier for educational applications. So the context of my work is science education, and one thing that can really help students is that their teachers are aware of the individual concept level difficulties and proficiencies of students. If you have a single number, the number we're familiar with, the percentile, a score, or GPA, that's good for keeping a record, but that's not really helpful in improving student learning. So what teachers typically end up doing, especially at the school level, is that they conduct assessments, they grade them, and then they analyze them. And this is something that most students are not aware of, is the fact that they're looking at those results and trying to figure out the concepts that students are struggling with or the concepts that they're actually good at. And this analysis is tedious, and in the context of public schools here in Washington, teachers don't have the resources or the time to just keep on doing this all the time. So what we looked at was trying to use machine learning and natural language processing to automate this analysis part. Text classification is a very common tool of natural language processing, has a lot of applications, and some of them you're all familiar with, such as spam filtering. And how this fits in with our work is we can build a text classifier that automatically finds the concept that each question is trying to assess, or multiple concepts, and then we can use the scores on those questions with these concept labels to figure out the concepts that students are struggling with or they are proficient at, and we can do this for every student. And another thing we can also identify using the system are those questions that every one of us here has experienced, which no matter how much you know, no matter how much you know, you have no idea what to answer for them. So here are a little bit of details about my work. So I had questions from 3rd, 4th, and 5th grade science, and they were divided into 8 topics. So here there are a couple of sample questions. The first one is from the topic of environment. The second one is from the topic of forces and motion. And 2 practical issues that I had building this classifier was, one, I had a really small amount of data to work with. And the second was, this, given the nature of the topic, some of them had a lot of overlapping vocabulary. So this is a picture, a very high-level picture of our system. So if you look at the top, that's the training part. So we took the questions. We represented the words in a question using counts on numbers. That's the part of feature extraction. And then knowing the topics for those questions, we trained our machine learning algorithm to be able to predict the topic for questions it had not seen before. And that's basically what happens during the testing phase. We take new questions, we do the similar feature extraction that we do at the training process, and then we predict the topic that that question is associated with. And the accuracy of a classifier is just how many times we get the predicted topic right for the set of questions that we haven't seen in the training process. 2 of the things that I'm going to talk a little bit more about is the process of feature extraction and the classifier itself. So questions are words, and it comes down to how we represent those words. So we could do simplest case binary, whether a word is there in a question or not, or we could do counts of number of times a word appears in a question, which was our baseline. And a bit more nuanced way of doing that is to represent each word as a vector in an n-dimensional real number space. And the idea is to learn this representation so that words that are closer in meaning are closer in this n-dimensional space. And this is actually a nice part of the whole system that I built, because for this I was able to use external data. So I took about 38,000 science articles from Wikipedia and used the words from those science articles to learn this representation for 50,000 words, which I was able to plug into the classifier. And this helped to circumvent the problem of having really limited training data for the classifier. For the classifier, as a baseline, I used a support vector machine, and that took a bag of words as input. And since it was just a count of words, the order of the words in the question wasn't accounted for. The second thing that I tried was a convolutional neural network, and what it basically did was it took filters of size 3, 4, and 5 words and swept it sequentially down the entire question. And since it was sequential, it did account for the order of words that appeared in the question. So here are some of the results that I got for both the word embeddings, which is the vector representation of words, and the accuracy. So if you look at the first table, there are 4 words, theory on, the number 7, and he, and listed below are the 5 nearest neighbors in the 128-dimensional space. And this representation I learned on the external data before training the classifier. So if you look at this table, and this 5 nearest neighbors are in terms of Euclidean distance in the 128-dimensional space. And this shows that we were able to learn some interesting structure in the way the words are related to each other. And when we plug this set of pre-trained word representation into our convolutional neural network, so if you look at the second table, we got an accuracy improvement of 11%. So we were able to get a 96% accuracy on the classifier that was classifying over 8 topics. And our baseline SVM also actually did really well with the 91% accuracy and took a lot shorter time to train. Both of these were a lot better than random guessing, which got us an accuracy of about 22%. So this shows that this is promising to be able to use for analysis of student learning. So thank you everybody for listening, and I want to thank everybody in my lab, Professor Bell and Deb from the College of Education, and also the people that I worked with Washington STEM and Mesa, and my original sponsors, Fulbright. Thank you.
So does anyone in the audience have any questions? Okay. All right, thank you. Next, we have Taylor Friett from the Noise Lab. So let's make some noise for him.
Hi, everyone. My name is Taylor, and I'm from the Noise Lab, which is led by Arka Majumdar. And today I'll be presenting on our optoelectronics platform in silicon nitride. So a lot of our work in our lab revolves around these optical interconnects, which are mostly useful over long-distance communications, as you can tell from the optical fibers that are laid underneath our oceans. whereas the electronic counterparts dominate for shorter scale devices. So there's this new emerging in-between area in data centers, and this is the rack-to-rack communications. Here, it's not so much a function of distance, but bandwidth that is pushing the transition from electrical interconnects to optical interconnects. And so what we do in our lab is we kind of address some of the needs that are particular to this use case. Namely, we make lower threshold lasers, faster modulators, more sensitive photodetectors, and such. And so the work that I'll be presenting today will be mostly covering the lower threshold lasing. To achieve this and interconnects in general, you need two components, generally speaking. This is the passive photonic circuitry and your active media. And so, unlike traditional integrated photonics, which use silicon as their photonic elements, we use silicon nitride. And this is largely motivated by the thermal optic effect, which is much larger in silicon. This is a very negative effect because it detunes your photonic elements as a function of temperature, which would be very problematic in data center environments where you have wild temperature fluctuations. However, silicon nitride, unlike silicon, is a very passive material. So to regain some of the functionality, we have to incorporate some of these new 2D materials, these atomically thin materials. And so one of the best things about these, besides their functionality, is that they're very easy to use. Unlike traditional materials where you have to use epitaxial bonding or epitaxial growth or wafer bonding, these ones you can just place on top of each other, much like Lego blocks. So you can make heterostructures very easily. And this is particularly advantageous because these materials span the entire bandgap region that you're interested in, namely from semiconducting to insulating, with many direct and indirect bandgap semiconductors in between. And so our work mostly focuses on one material in particular, which is a direct bandgap semiconductor named tungsten diselenide, or WC2. So, of course, our first experiment was to try to make this material laze. And so to do that, you need a little photonic cavity. This kind of recirculates light, and it couples to this 2D material, so, which was easily placed on top. And so we were able to see ultra-low threshold optically pumped lasing with this device. The unfortunate part is that the cavity was gallium phosphide, which was motivated by our lack of silicon nitride cavities at this time. So we've begun to construct some of these devices. And so here I have a disk resonator made in silicon nitride. So unlike the photonic crystal cavity, which binds the light using these hole-like structures, this one binds light by having it circulate through. And then you address this device through these gratings. So this is sort of the response of this device. And what's important here are these narrow dips. These are the actual optical modes that we're measuring. And so if you want to have ultra-low threshold lasing, one of the most important things is to have high-quality resonances, which we've shown. So just like our last device, we incorporated some tungsten diselenide on top. And so this is the spectrum we see once we excite this with a Heine laser. This broad background here is just coming from the WC2, and these bright peaks are coming from the cavity enhancement. And so what we were hoping to see is that one of these peaks would dominate and become much brighter and much more narrow than all the others. Unfortunately, we didn't see that, and so we're still looking for lasing. The other improvement is we need to make sure that these things are electrically pumped and not optically pumped. Optically pumping is not conducive to real-world environments. And so we made these heterostructures for electroluminescence. And so here on top is a diagram. This basically shows our tunneling junction electrodiode. And down below, you can see the optical micrograph of such a structure. And then to get better enhancement, we put an optical cavity on top, and then this is what we saw when we pumped it. And so the blue curve is the improved curve, and the red curve is just the old EL signature. And so by pumping harder and harder, we were able to get better and better efficiencies. And so hopefully we'll be able to implement this in silicon nitride as well, and perhaps see electrically pumped lasing. Thank you for your attention. If you have any questions about our work or other work that we do, please feel free to contact either Arca or I at these emails or view our website.
Any questions for now? Okay. Thank you. Next, we have Jimin Kim from the Data-Driven Dynamical Systems Lab.
Thank you. Good morning. My name is Jimin Kim. I'm the graduate student here at the UWEE department, and today I'm going to talk about the neural interactome. The neural system consists of two fundamental layers. The first layer is a static mapping of the network connectivity called the connectome, and the second layer is a dynamic biophysical processes between neurons, i.e. the neural dynamics of the network. And in the recent year, there has been a significant progress resolving and modeling both of these layers for the different biological organisms. And such progress provides computational basis for us to build a model which incorporates both static and dynamic layer for different neural networks. And such model is called a dynome, as it corresponds to the dynamical system on top of the static connectome. So for the efficient studies of the such model, I developed the neural interactome framework, which is the interactive visualization platform for the neural networks. The framework has the neuron stimuli panel, which enlists all the neurons in the system, categorized into different group type, and each neuron is a click of a button where you can either inject arbitrary stimuli or remove that neuron from the system to perform the neural ablation. On the right side, we have the graph representation of the network, which visualizes both structure as well as the real-time neural responses of the network. So for here, the nodes correspond to the neurons, whereas the edges correspond to the connections between the neurons. So for displaying the real-time neural responses, the larger the membrane voltage, the larger the radii, and the sign of the voltage determines the color of the nodes. So I applied this framework to the nervous system of the C. elegans worm, where its connectome has been fully resolved from the serial section electron microscopy, and its computational model describing the biophysical process between neurons has been recently introduced. So here I show one of the experimental scenarios that can be tested with the neural interactome, and in particular, I focus on the subcircuit of the network associated with the posterior touch response. So in the experiment, this circuit is known to drive the forward locomotion of the worm. So I'm just going to play this video here. So on the panel, I have stimulated the relevant set of neurons. And as I click the play button, we see that the network starts displaying the corresponding neural dynamics in real time. And if you pay attention to the left side of the graph, where all the motor neurons are concentrated, we see that the stimulation induces a strong two-mode oscillations between these neurons that actually drive the forward crawling motion of the worm. What I can also do is I can add the additional stimuli on top of the existing stimulation, and we see that the network also responds to such dynamic modification of the stimulation in a real-time manner as well. So for this particular example, adding this sensor neuron, which is in the experiment actually associated with the oxygen sensing, actually suppresses the ongoing oscillation of those forward motor neurons. So here I focus on the different subcircuit associated with the anterior touch response. So in the contrast to the previous scenario, this circuit is in experiment known to modulate the backward locomotion. So on the panel, I'm stimulating the completely different set of neurons this time. And similarly, as I click the play button, notice how the neural dynamics now involves a lot more neurons than that of the forward crawling scenario. And what we can also see is that the dynamics now activates the different groups of motor neurons that are more involved with the backward crawling rather than the forward crawling. So this type of visual characterization really shows how different the backward locomotion dynamics is to the forward locomotion dynamics. What I can also do is I can pause the simulation and click on the time bar anywhere I want to travel back into the previous time point of the simulation. And I can also zoom into the particular subcircuit to investigate further on the local behavior of the neurons of my interest. So in the summary, the neural interactome is the interactive real-time visualization of the neuronal system. And with its novel methods, the framework significantly simplifies the studies of the dynamic network model and therefore assists in the discovery of the unique network functionalities. And subsequently, it has a potential to be used as a platform as a pre-experimental tool for mapping out the scenarios to be explored empirically. And it can also be applied to the other neuronal system, ranging from the biological systems such as the Drosophila medullae, which is the fruit fly, mouse retina, mouse primary visual cortex, which have been recently resolved in the Allen Brain Institute, and also the artificial neural networks, such as the recurrent neural network. So I'd like to thank my collaborators here and my departments for this project, and thank you for your attention.
Any questions? Okay. So last but not the least, we have Astrini C from the Biorobotics Lab.
Thank you. Hi, everyone. My name is Astrini, and today I'm going to talk about the Smart Grasper. So the research motivation behind the Smart Grasper came from a minimally invasive surgery, or MIS, and robotic surgery in general. So in MIS, there are 3 incisions made on the abdomens of the patient, one of which is for the camera and 2 for surgical instruments. Among these 2, one of them is typically used for a surgical grasper. So in MIS, surgeons interact with tissues, they perform operations and procedures using all these elongated instruments, and also remotely from a console in robotic surgery. So the big question that we have is MIS better or more beneficial than the traditional open surgery? The answer is yes and no. From the patient's perspective, MIS is more beneficial than the open surgery. There is less incision, which means less scarring, reduced blood loss, as well as faster recovery time and shorter hospitalization period. But on the other hand, MIS is not as beneficial from the surgeon's perspective. There is no direct contact between the surgeon's hands and the tissue, which means they're not able to fill different tissue types and different tissue conditions. So most of the times, the surgeons will unintentionally apply too much force that will end up damaging tissue. A lot of methods or ways to address this problem is by introducing haptics so that the surgeons can feel again. Our approach to addressing this problem is slightly different. We would like to address this problem by answering these questions. Can we build a smart surgical grasper that is as good as the surgeon's hands? Can we build a smart surgical grasper that is capable of performing tissue classification? Can it identify different tissue types, distinguish between one tissue type and another? Can we use such a smart surgical grasper to perform an exhaustive tissue property characterization? Imagine a complete tissue property database that can be used for a realistic surgical simulator. Can we also use a smart surgical grasper to perform tissue diagnostics? Our lab is building a smart surgical grasper that can be shown in the following figure. So on a grasper handle, we have strain gauge and encoder for measuring tissue force and displacement. There's also a motor for applying specified amount of forces to tissue. And on a grasper jaw, we have 3 sets of sensors. The first set, we have LED and photodiode for measuring tissue optical properties. We also have a thermistor for measuring temperature. And finally, we have an electrode array for measuring tissue electrical impedance. The way the grasper works can be summarized in the following block diagram. So first of all the sensors will collect data from tissue, and these data will be sent to a computer. The computer will process all this measurement data and come up with a motor command that will be sent to the grasper. So using this grasper, we collected animal data on pigs for 4 different types of tissue, which are liver, small bowel, bladder, and gallbladder. So in these experiments, we collected force and displacement data from the tissue. And using this force and displacement data, we ran an extended Kalman filter algorithm on a dynamical tissue model to estimate tissue elastic properties, which are the alpha and beta shown in the equation on the board. And using the estimated values of alpha and beta, we then calculate the estimated force values and plot it versus displacement on the following curve. And we can see that there are 4 distinct curves, which represents 4 different tissue types. So this means the smart grasper, as well as the algorithm, is capable of performing tissue classification using only force and displacement data. So moving on from this, what we'd like to do is perform a more robust tissue classification using the 3 sets of sensors at the grasper jaws. We'd also like to perform tissue diagnostics, which include differentiating cancer tissue from healthy tissue, and also measuring tissue oxygenation level, which is pulse oximetry, as well as applying a closed-loop control for the force that is applied to tissue so that there's no injury can happen on tissue. So with that, I would like to conclude, and I would like to thank my team members for their tremendous help and contribution in this project. Thank you.
Awesome. Thank you to everybody for staying until the end, and thank you to all the participants for the really interesting talks. Before we end, GSA would like to thank Professor Mari Ostendorf and Brenda Larson for their continued support and advising, Professor Matt Reynolds, the Colloquium Committee, and the event staff for allowing us to use the time for the colloquium, and the chair of the EE department, Professor Radha Puvendran, Brian Crockett, and Tina Montgomery for advocating for the travel award. So, whoops, sorry. Here are all the participants that spoke today. Please vote for your favorite speaker at the Catalyst link on the top. Thank you again for coming, and please have a great day.